I get to talk about some of the work which I probably talk less frequently about, and that's going to be non-swine influenza viruses in the swine host. So we won't get to see a lot of busy dendrograms that we usually look at with our swine influenza viruses. But some of this work I think will be interesting to you here in the Midwest because of the outbreak of the H5N2 high-path avian influenza virus and wondering what's going to happen this fall. And then we also took a look at the new Canine H3N2 that emerged also earlier this year in the U.S. Okay, so just to get us all up to speed with influenza A virus, there are a number of hosts which can be infected with the influenza A virus, but the natural reservoir are wild waterfowl, and all of the subtypes of the hemagglutinin and neuraminidase, except for those that are restricted to being found in bats, can be found in different combinations of in the wild waterfowl. Occasionally we have spillovers from the wild waterfowl, more commonly to poultry, but occasionally to humans and swine. But for mammals, we tend to have influenza A viruses that become host restricted with less common spillover. But we do have a specific niche here between pigs and people and pigs and turkeys, and we also know we've got this spillover from wild waterfowl to turkeys and chickens as well. We'll also briefly mention the Canine H3N2 later in the talk. So a number of H5 viruses were identified in U.S. That were identified to be highly pathogenic, and we know that those spread from wild birds over on the Pacific into the Midwest, and so the question began to be asked, what will happen to the swine population? Are they at risk to these H5 viruses? And this was a new introduction of an HA that was seen in Europe and Asia but hadn't been seen in the U.S. So going back to some previous work, there's been a number of H5 strains, both high path and low path, which have been used in experimental settings to see if pigs could be infected with these viruses. And in general, pigs tend not to be easily infected with avian-adapted H5 viruses, no matter whether they are high path or low path. And high path is really just a term that is applied to how the virus behaves in poultry, not necessarily how it will behave in any other species. But from a number of studies that we have done in ADC, when we infect the pigs directly through the respiratory route, we can see some low-level replication, mostly in the lower respiratory tract or the lungs, with minimal nasal replication, minimal shedding, and very limited evidence of transmission from infected pigs to naive contact pigs. However, there was a seminal paper many years ago by Kita et al. That demonstrated the reassortment potential even of these low-level infections of avian viruses in pigs. And so that goes along with the mixing vessel theory for swine, and so this has hung over the head of swine researchers and swine producers since that time. However, no H5 viruses are endemic in pigs anywhere in the world, although there are reports occasionally of serial positive pigs to H5 viruses and sporadic virologic detections of H5 viruses, mostly in Asia. So this is a bit outside my expertise area, so I see there's a few people in the room who can probably talk lots more about this. But stealing some slides from the USDA APHIS website, these are the early detections in wild birds of these H5 Eurasian lineage high path. And they were detected with different neuraminidase, so that's why there's the H5NX there in the title. And so early on we had these isolations, northern pintail, mallard, widgeons, a green-winged teal, and then we've got H5N2s, H5N8s, and then this H5N1. So this is what was happening over on the Pacific Flyway. Okay, so then we had some poultry cases beginning in late 2014, and then continuing on to 2015 from the Pacific Flyway, and then into the Mississippi Flyway in March of this year. All of these isolates that were found in the Midwest and the Mississippi Flyway have been the H5N2 subtype. So why was there concern for pigs and high-path avian influenza? I think it'll be pretty obvious to those of you here, but most of the cases in poultry, so in commercial chicken and turkey flocks, or here in the Midwest, similar to the highest population density of swine, and so if you overlay those together, then the question becomes obvious with all this density of infected poultry flocks, what risk does this have to the swine industry? So what we did at the NADC in partnership with NVSL and our Southeast Poultry Research Lab down in Athens, Georgia, is we took 4 strains of these high-path avian influenza, a turkey Minnesota isolate of H5N2, an earlier isolate from a northern pintail H5N2, and then we also looked at the H5N8 and the H5N1. And so we experimentally infected 15 pigs, our primary group here, and looked to see whether there was going to be any replication in these primary infected pigs. And then we added contact pigs 2 days later to evaluate whether we could have transmission from the primary infected pigs to these naive contacts. So to look for nasal shedding, we do nasal swabs on days 1, 3, and 5 post-infection, and in the contacts at 3, 5, 7 days post-contact. And then we also euthanize subsets of these primary pigs on day 3 and 5. And then we look for seroconversion at day 21 and 19 in subsets of the primary and contact pigs. We also took advantage of some of these primary infected pigs and wanted to collect some positive control anti-sera. So we had to boost 2 of each of these virus group pigs from these virus groups to generate reference anti-sera. Because as you will see, we had poor seroconversion. So this is the data slide, and it's not very interesting, which is good news. We had the H5N2s up here, the H5N8, and the H5N1. The top line under each of the viruses would be the primary infected pigs, and then the contact pigs. So for the nasal swabs or nasal shedding, we had mostly negatives here. We had 1 primary pig, which we could detect virus in the nose at day 5. All of the contact pigs, though, were negative by nasal swab. However, when we looked in the lung tissue on the necropsy days, we do see some low-level replication in subsets of the pigs. So we're just looking at the primary infected pigs here. The number on the left side of the hash would be the PCR positives, and on the right side would be the virus isolation positive. So we do see some virus replication deep in the lung from these H5N1 viruses from the U.S. However, when we look at seroconversion, and this is based on the hemagglutination inhibition assay to the homologous challenge strain, even though we have some replication in the lung, we only had 2 of the 5 in the H5N2 groups, and none of the pigs in the other virus groups seroconverted by HI. We are still planning to run the MP ELISA, but that hasn't happened yet, so that's still pending. So this is good news for us, but I don't think it's time for us to turn a blind eye or go to sleep, because there's a number of different genetic clades of H5 viruses, and so I apologize for the MAC transversion here to PC, so you can't see the strain names. But these viruses that we just looked at experimentally, these are the North American H5N1. They belong to a clade called 2.3.4.4. However, some work that my group had previously done was looking at this Euro-African lineage of H5N1s from a clade called 2.2. And these were of interest because they were associated with a number of human cases in Iraq and Egypt, the Middle East, and then this clade went on to evolve into the clade of viruses that Egypt is still struggling with. So this was a reassortment study where we took the H5N1, highly pathogenic avian influenza. This was work done in collaboration with the CDC, and they generated a reassortant virus that had the internal genes from the pandemic H1N1. So surface genes of the H5N1 and internals from the H1N1, which we all know is influenza that's shared between people and human. And this internal gene machinery is quite efficient in the pig host. And I'll just mention this work was done prior to the moratorium and federal funding pause on gain-of-function research, and this work is not published because of those two things. So we had 4 different virus groups in this study, but I'm just reporting the two H5N1 viruses. This is the parent virus, so the whole H5N1, and this is the reassortant reverse genetic derived virus that has the internals of the pandemic H1N1. So the black line here is the nasal shedding of the parent virus, and the gray will be the nasal shedding of the reassortant. But what's important here to note is the hatched line here, that would be detection in the contact pigs. So this is very different from what we saw with our current H5 viruses, is that we were seeing some transmission and nasal shedding in both the primary and in the contact pigs. And then when we look at the seroconversion in these two groups as well, we see the parent H5N1 not being transmitted to the contacts and being detected by seroconversion very efficiently, but when we use that reassorted virus with the pandemic internal genes, the virus was much more efficient at transmitting to the contacts. So this is not meant to cause unnecessary worry because this is a different lineage of H5 viruses, but it is something I just want to point out that if these H5 viruses we currently have the opportunity to reassort and pick up a different internal gene combination, we may be looking at something different than just the parent viruses alone. So we also looked at the risk for this new canine influenza virus, this H3N2, as to whether that was going to be a risk for swine. Obviously, this is a similar subtype, but a very different genetic lineage than our own swine H3N2s. This H3N2 is genetically and antigenically distinct also from the previous canine influenza virus, which was an H3N8 of equine origin, spillover from horses to dogs. So this H3N2 is an avian lineage virus, but it was first identified in dogs in southern China and South Korea around 2006. And it was responsible for a larger outbreak in the Chicago area in March of this year, and then disseminated from there. It's more commonly associated with these community animal shelters, boarding facilities, daycares, and dog parks, and doesn't seem at this point to be widely disseminated in the dog population in the U.S. And if we look at where these outbreaks have occurred in the U.S., they tend to be associated more with urban areas rather than the pattern that we saw with the Mississippi flyway and the wild waterfowl avian influenza spread. So a similar study design that we did with the H5N1 viruses, again, we infected primary pigs, and then 2 days later placed the naive contacts, looking for nasal shedding in the primaries and then transmission to the contacts. We necropsied at day 5 on the primary pigs to look for evidence of replication in the lung and lung lesions, and then at the end here we're looking for seroconversion in both our primaries and contacts. So I don't have a data table here because it's basically not interesting at all, but we had, again, mild to no lung lesions. We had low levels of virus that we could detect in the lower respiratory tract of the primary infected pigs, but no evidence of transmission from the primary to the contact pigs, either by nasal swabs or seroconversion. So take-home points, there were 4 U.S. high-path AI isolates that we used in our swine studies, both from the late 2014 and 2015 wild bird and poultry outbreaks, and then the Canine H3N2 from 2015. Pigs were directly inoculated with high dose of virus, and then naive contacts were placed, and our findings so far indicate that pigs directly inoculated with these viruses have low-level replication deep in the lung, but no evidence of nasal replication or transmission from the infected pigs to non-infected contact pigs. Another not positive note that we want to emphasize is that the infection in the primary pigs of all these viruses were detected by the RT-PCR screening test that the diagnostic labs would be using. So if there was a rare event where these viruses did spread to pigs, our diagnostics labs would be completely capable of picking them up. We do have some seroconversion in the primaries by HI, and so since H5s don't circulate in pigs, this could be a strategy to look for this virus in pigs. However, the lack of seroconversion in some of our primary inoculated pigs means that further work is required to determine how to sample and the reliability of the HI. We will be doing the ELISA on these pigs. We had some problems with getting samples out of our BSL-3 lab with some of the new tighter regulations, and so that got put on hold, but we will get those done soon. However, the cautionary note here is that not all high-path H5 viruses are equal, and we have previous data that showed that the clade 2.2 lineage had some transmission that it was increased by switching out those internal genes to put the H5N1 on the pandemic H1N1 internal gene backbone. So there is that nagging question of what if these North American H5N2 viruses reassorted to acquire swine-adapted internal genes, such as our trig swine lineage or the pig, the pandemic internal gene lineage. And if you're thinking about turkeys the way I think about turkeys, that might be something that is a concern for you as well. However, most avian H5 and H7 viruses that have been previously tested in pigs fail to sustain efficient replication and transmission. So the risk is not zero, but it seems to be unlikely and would be probably self-limiting or require multiple mutation or reassortment events for this risk to occur in the swine population.